In question 62, we're told that a student used a light microscope at a magnification of 40 times to observe a slide of non-dividing tissue. In cells of this tissue, we're told the student noted the presence of a large central vacuole and a cytoplasm containing a single stained round structure, which we've modelled here. We're then asked, out of these three options, which of the following could have been seen by the student in this tissue at this magnification of 40 times. So we have a cell wall, we have mitochondria, and we have chromosomes. Now, this is a bit of a tricky question, and the best way to think about this is to think back to the times that you looked through a light microscope in your previous studies. You'll have encountered plant cells before in your previous studies, and you will recall that you should have been able to see a cell wall as well as a vacuole and a nucleus. Now, that helps us with our first part of the question, but it does make it a bit more ambiguous when it comes to assessing whether we can see mitochondria. You may have been able to observe, perhaps at 50 times magnification during your previous studies, individual structures within the cytoplasm which you may or may not have been able to identify. Some of these may have been mitochondria, but since you are unable to identify them at this magnification, it's not necessarily right to say that you can see mitochondria at this magnification. So we can say this is wrong, and of course we know that chromosomes must be wrong, since when it comes to DNA and its associated histone molecules, these are much, much smaller than anything that can be observed with a light microscope. So we can say that our answer must be that only the cell wall can be seen or readily identified, and so our answer is B.